Could a common blood mutation be the key to preventing Alzheimer's disease? Imagine if a tiny change in your blood could hold the secret to staving off one of the world's most feared diseases, Alzheimer's. It may sound far-fetched, but that's exactly what a groundbreaking new study is proposing. Blood stem cell mutations, once mostly seen as a danger, might actually have a silver lining when it comes to brain health. But here's where it gets controversial. Not all mutations are created equal, and some might even work in our favor. A fresh investigation featured in Cell Stem Cell unveils an intriguing twist. Some genetic mutations in blood-forming stem cells could lower the risk of developing late-onset Alzheimer's disease. This research, led by Baylor College of Medicine, throws a spotlight on a gene called TET2. Both human participants and mice with mutations in this gene seemed less susceptible to Alzheimer's, while another gene, DNMT3A, didn't offer the same promise. If confirmed, this could pave the way for entirely new strategies to prevent or delay Alzheimer's disease, an illness that robs millions of their memories every year. Dr. Catherine King, a leading researcher from Baylor and Texas Children's Hospital, has spent years delving into the mysteries of blood stem cells, formerly known as hematopoietic stem cells. These crucial cells reside inside bone marrow and are responsible for producing our body's entire supply of blood cells, including red blood cells that transport oxygen, immune cells that fend off illness, and platelets that help stop bleeding. Here's the surprising part: as we age, it's common for blood stem cells to acquire mutations. Up to 1 in 5 people over 70 have them. While most of these changes go unnoticed, sometimes a mutated cell multiplies faster than its neighbors, creating a growing population, or clone, of altered cells. This phenomenon, called clonal hematopoiesis, has always been a double-edged sword. On one hand, it's been strongly linked to serious conditions like heart disease, stroke, blood cancers such as leukemia, and lung disease. But what about its connection to Alzheimer's? Is it possible that this process, feared for its risks, could sometimes protect the brain? And this is the part most people miss. The Baylor team decided to dig into 2 genes, TET2 and DNMT3A, that are most often mutated in clonal hematopoiesis. These genes are also tied to inflammation, which plays a major role in Alzheimer's disease. To get answers, the scientists combined large-scale human genetic data from the UK Biobank with experiments on mouse models engineered for Alzheimer's. What they found could spark heated debates in the scientific community. Only the TET2 mutation, not DNMT3A, was linked to a striking 47% reduction in the risk of developing late-onset Alzheimer's in humans. In laboratory mice, transplanting bone marrow with TET2 mutant cells helped slow memory loss and reduced the formation of harmful beta-amyloid plaques in the brain, a hallmark of Alzheimer's. DNMT3A mutations, by contrast, showed no such benefit. How exactly does this work? According to Dr. K.D.A. Matatal, the study's first author, immune cells that originate from TET2 mutant blood stem cells are better at traveling into the brain and cleaning up beta-amyloid buildup. It's this combination, more immune cells reaching the brain and each being more efficient at removing disease-causing debris, that appears to explain the protective effect. This idea flips much of what we thought we knew about blood mutations on its head. Until now, scientists mainly viewed clonal hematopoiesis as a negative factor. Dr. King notes, this is the first time we've shown these common mutations can have such different effects on disease, some harmful, others protective. We need to shift our thinking and look at each mutation individually, weighing both their risks and possible benefits. The implications are huge. Not only does this establish a new way to study how blood cell mutations impact Alzheimer's, but it also hints at ways to potentially shield the brain from degeneration before symptoms even arise. Could future therapies deliberately harness or mimic the TET2 mutation's effects? But here's the million-dollar question, and it's bound to spark passionate opinions. If certain blood mutations can guard against Alzheimer's, should we be seeking them out or even engineering them? Or do the potential risks to heart health and cancer outweigh the possible brain benefits? Where do you stand? Share your thoughts, and don't hold back. Do you think the promise of protection from Alzheimer's justifies rethinking the way we view blood mutations? Or is it too risky to meddle with our genetic code? Let's discuss.